Hello and welcome to this video on one of biology's favourite creatures, Drosophila, or as it's otherwise known, fruit flies. Fruit flies are a plague in orchards, homes and greengrocers. In a laboratory they can be extremely useful, if not powerful as a research tool. While not as infamous as rodents, fruit flies can be commonly found in research facilities working on genetics, heritable traits, physiology, anatomy, and things like embryology. As far as what they do, they're generally used as a model of life, but they can simultaneously be very far off the mark. Despite that, we need to start somewhere, and generally speaking, fruit flies tend to be fairly reliable in that respect. It's why Drosophila is so useful. They are small, easy to maintain, and plentiful. As we also know, the whole genome, it's relatively simple to modify it in specific ways to recreate all kinds of phenomena. These adventures in science led to the discovery of genes creatively given names like Kenny that causes Drosophila to die randomly, or the cheap date gene that makes them more easily intoxicated by alcohol. And don't think that fruit flies or Drosophila are a relatively new phenomenon either. Their first documented use goes back to 1901, all the way back at the start of the 20th century, quite literally, where researchers in Harvard used them to, find a whole bunch of things, but mostly looking at inheritance traits. This led to one of the earliest gene names, the white gene, which is related to the colour of the Drosophila eye. Normally it's a pinkish red colour, but with certain genes that can be inherited, it can turn white. This is why, for more than 100 years, the very low cost, rapid generation time, and variety of genetic tools we have available have made them almost indispensable as a basic research tool, for lack of a better word. In fact, the variety of molecular research tools we have available now mean that they're even more useful than they were previously. The modern era, let's say, of Drosophila or fruit fly research really only took off when we were able to get into the nitty-gritty details of the genetics, and this has allowed various fields of research to take greater advantage of them. Most notably, there have been Nobel Prize winners who relied on them solely to get that prize. There's also the fact that, much like with rodent research, Drosophila does have certain genes that are almost perfect homologs of those that are involved in human development and diseases. It means that we can, in theory, play with those genes in the fruit fly and see what happens, and have a good idea that we can make the same changes in a human and observe ideally the same outcomes, but at least we know how to make those changes even if we don't know exactly what will happen. Like a lot of small annoying creatures like butterflies and moths, Drosophila has a fairly straightforward life cycle. It involves going from an egg to a larvae into a pupa and then the fruit fly itself. Once it's fertilized as an egg, the embryo will develop within it for about 24 hours, though this can take longer at colder conditions and be shorter in warmer conditions. Once it hatches as a larvae, it will eat and grow. It will molt 3 times as a larvae until it becomes a pupae. At this time it will undergo metamorphosis into the adult fruit fly, and that will take roughly 4 days. During metamorphosis, most of the embryonic and larval tissue is completely destroyed, leaving only the adult tissues behind. These are things like wings, legs, and eyes. What's important is that these develop from specific known cells called imaginal discs. These very specific sets of cells have been present since early embryonic development, but they have been specifically in dormancy until the fruit fly starts developing into its adult stage. These features are why it is especially useful as a research tool in embryology, because it has so many distinct and very clear stages that make it very easy to figure out what's going on, as long as you can identify which of those imaginal discs you need to look at a given time and find a way to isolate them. One thing that we should mention is that, although they're called a fruit fly, they're not actually necessarily feeding on the fruit itself. Rather, it feeds on yeast, and one frustrating thing about the fruit fly is that it will actually spread yeast from place to place. This is one reason why vineyards, breweries, and similar really don't want them, as it can lead to contamination. the fruit fly will eat the yeast growing on the fruit or anywhere else for that matter. In the early days, fruit flies were kept in bottles containing something like a banana mush, although that was kind of questionable and honestly not ideal. Nowadays they use something closer to an agar, although not quite. It's somewhat more complex than your standard agar mix, as it involves water, rather obviously, a cornmeal, yeast, soy flour, malt extract, a corn syrup, and the agar itself, which frankly is getting closer to a beer recipe than it is a, agar solution. The reason why you need such a specific cocktail of ingredients is that you need the agar present to harden the top of the, let's say, feeding solution, so that the fruit flies don't sink into it or get stuck, but it needs to be soft enough that the larvae can crawl around and feed on it without any issue. The other thing you need to do is have some sort of semi-permeable membrane over the top of your containers holding your fruit flies. This is generally done with something like a foam or cotton wool. It prevents the fruit flies from escaping, but it also ensures that, nothing can get in there. One reason you want to ensure they don't escape is so they don't annoy you, but the other reason is that if they're genetically modified, you don't want them escaping and breeding with others that aren't genetically modified. At the end of the a lifetime that the fruit fly is needed to grow and be alive, they're generally euthanized using something like carbon dioxide. Fruit flies have contributed extensively to things like our understanding of regenerative medicine. That is because of how they grow, and particularly those imaginal discs. They are especially important given that what is effectively an adult organism starts growing again almost as though they were going through embryosis once more. Because of this, the pathways and processes that occur effectively undergo what we would consider tissue regeneration for an adult now, and that helps us in understanding things like cell division, or what happens when cells don't divide properly, and how to ensure that, the right things happen in the right places at the right time. Those imaginal discs have been the center of focus for over 40 years. If you take the same discs from a developing pupae and you put them into an adult fruit fly, they'll proliferate, but they won't differentiate. By contrast, however, if you take the same sort of imaginal discs from one larvae and put them into another, they will do exactly what they're meant to do. They will proliferate and they will differentiate. If, however, you take the same set of imaginal discs and you repeatedly transplant them between different pupae, you'll find that they can end up differentiating into anything almost, and this is called transdermatism. If you take the cells that would make an imaginal disc in an adult fruit fly and you put them into a developing fruit fly larvae, you'll find that the cells have differentiated enough that they will regrow and turn into exactly the same predictable cells every time in the developing larvae, allowing you to regenerate or duplicate tissue. That alone isn't the only use for Drosophila. They are also incredibly useful when we're talking about drug discovery. Not just because they can be genetically modified to have the same sort of problems that a human would have, but that we can look at the biochemical pathways within Drosophila to understand, theoretically, what sort of effect a drug might have on the same sort of conserved genetic pathways between the Drosophila and human. This does arguably remove the need at this point in time of drug discovery for things like a rodent model, rodents which can be very expensive, very difficult to handle, and take much longer to develop, versus a fruit fly which may take up to a week to grow. This makes them very desirable if you're trying to test large numbers of, let's say, plant extracts or other bioprospecting drug options, or possibilities more accurately, instead of trying to do things like cell culture, or trying to grow something, for example, like a rodent or similar. They are much easier in that respect. There's also the, to a certain extent, arguments for and against the role of biological modification or bioengineering using relatively unknown and unpredictable changes within Drosophila. And this again owes to the fact that there is a degree of homology between the fruit fly and the human, because you can take a gene and put it into the fruit fly or otherwise modify the fruit fly's genes, you can in theory see whether or not altering something within them could have an adverse effect due to environmental conditions. For example, something that might affect their survivability under certain light conditions, heat, cold, water exposure, and so on. Or if you want to see whether or not there are toxicity issues from certain things. All of these can be done in a fruit fly to some extent. Given the nature and functionality of this sort of testing, it would have been incredibly important and useful to have had them do this with something like the thalidomide scandal, where they would be able to see that during embryogenesis, certain things weren't developing when exposed to those drugs at certain times. The strength of using a fruit fly model over that of cells is, well, cell culture tends to be a single cell culture, and because of that, it doesn't necessarily have the necessary complexities of a whole organism, whereas the fruit fly is, well, a full organism. As such, you can look at what happens not only at a single point in time in a single cell, but across the entire lifetime of the fruit fly, and arguably multiple generations. This also brings us to the role they have to play in genetic modification, or more accurately, in genetically modifying the fruit fly to have the same traits and consequently the same diseases as those who have either challenging or rare diseases, that creating a rodent model is, at present, fundamentally beyond the reach of science. This can either be due to the sheer cost of developing that model, or that the model is incredibly difficult to try and create in something like rodents. This same approach can also be used if a known disease, rather than being just what's known, is a rare or unique mutation of that disease, which again would be much harder to recreate in something like a rodent model or even worse yet primate model. As such, the fruit flies are a very useful tool. We earlier mentioned some of the more entertaining names that have occurred with, well, genetics in general, but a lot of them have come from fruit flies, like the Kenny gene. But the conventional naming scheme is where this gets at least somewhat more entertaining, let's say. The whole idea between fruit fly genes and naming is that you name them for what you first observe. So for one example, one of the genes led to a more pronounced, almost eyebrow-looking features, and so the gene was named Groucho, after Groucho Marx, a film star of kind who had similarly large eyebrows. Then there were the tribbles, where cells divided in a different way, very similar to those that occurred in Star Trek. And there are the hedgehog mutations. These are where the larvae are round and have extra bristles, similar to Sonic the Hedgehog. However, this is only true of those that have been observed in fruit flies first. If instead the mutation or trait is observed in another species and then found in fruit flies, the naming convention found for other species, which is generally going to be something rather boring and tasteless, honestly, better to say not entertaining, it tends to get attributed to the same gene within fruit flies. So this can be something like, for example, SPT516. Not exactly the most entertaining of names compared to something like Sonic the Hedgehog, Notch, or Tribbles. The fruit fly, or Drosophila melanogaster, is a well-established and very versatile tool as a model in biology. There aren't a great deal of models that were developed at the very start of the 20th century that we are still using for exactly the same purposes today, and certainly none that have been as adaptable and able to keep up with modern needs as much as Drosophila have. The species has found its way into things like biomedicine, developmental biology, growth control, gut microbiome research, innate immunity research, behavioral research, and even neuroscience. That is how widespread it is. The fact that there are multiple kinds of Drosophila species has also allowed for comparative research into these different species, and what they do at different times of their life and under different circumstances. For example, the development of sex chromes in genital shape, adult pigmentation, the other features that are effectively key and central to Drosophila to do what they do, how they do it, and in the way they do. It's also allowed us to create very specialized versions. Knowledge that goes beyond just what's occurring at a basic level, which is what we do with a lot of species. We describe what is happening. Rather, this has allowed us to have a great deal more depth of understanding of things like the kind of pathogens that predate on them, how they diversify as a species and how they behave, closer to how an anthropologist might study human culture than how a biologist might describe how cells react. They sit somewhere in the middle of these two. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.